Welcome to another session of the Pen and Paper Room. Today we will be examining why exercise-mediated increases in PGC1 alpha are impaired in obese individuals, and both of them have similar exercise characteristics. Let's first take a look at the mechanism through which PGC1 is activated. So we have ATP being broken down to ADP, then AMP. AMP then activates AMPK. AMPK then directly phosphorylates PGC1 alpha. We also have calcium being released from the sarcoplasmic reticulum, which will activate CAMK, which then phosphorylates P38 and in turn phosphorylates PGC1 alpha. CAMK also activates LKB1, which activates AMPK. AMPK also activates NAMPT, which converts NADH to NAD+, which is a substrate for SIRT1, which then causes the deacylation of PGC1 alpha to an active form of PGC1. Epinephrine also binds to beta-adrenergic receptor, which allows the activation of G protein for the conversion of ATP to cyclic AMP, which then activates pKa, which in turn directly activates SIRT1. Active PGC1 then induces transcription. We will now look at the difference in epinephrine levels between obese and lean individuals. Trained athletes have lower epinephrine levels, and if anything, lean individuals would also have lower epinephrine levels. This would result in less activation of beta-adrenergic receptor. This would then result in less ATP being converted to cyclic AMP. This would result in less activation of pKa, which would then lead to less activation of PGC1 alpha. The ultimate result of all of this would be a decrease in mitochondrial biogenesis, showing that epinephrine is not the answer to our question. Trained athletes have a greater amount of mitochondria, resulting in a lower ADP accumulation in the muscle. So if not equal, lean individuals would have a lower ADP accumulation. This would then result in less AMP, then resulting in less AMPK activation, resulting in a decreased phosphorylation of PGC1 alpha. The result of this would be a reduction in mitochondrial biogenesis, showing that AMPK is not the answer to our question. When exercising at the same absolute intensity, calcium levels do not seem to be different, meaning that CAMK will be equally activated, P38 will be equally phosphorylated, and PGC1 alpha will be equally activated in both groups. This then results in an equal mitochondrial biogenesis between both the obese and the lean group, meaning that CMK is not our answer. Based on the question, we know that lean individuals have an increased active PGC1 alpha, as well as an increase in PGC1 alpha protein, meaning we have an increased translation. But transcription must also be much higher since PGC1 alpha mRNA is also higher. So we know that it's not due to the activators, So
after examining these activators in the PGC1 alpha mechanistic pathway, they do not seem to be the reason for the difference in active PGC1 alpha in lean versus obese individuals. We hypothesized that the levels of RIP140 may be elevated in obese individuals. RIP140 inhibits transcription of PGC1 alpha. More specifically, RIP140 forms a complex with PGC1 alpha and results in inhibition of PGC1 alpha mediated transcription. We hypothesized for the reason that the decreased level in the active PGC1 alpha in the obese individual could be due to an increased level of RIP140, which would inhibit PGC1 alpha mediated transcription and thus decrease the inflow to the PGC1 alpha mRNA pool.
Now let's look at what the literature says.
This study aimed to evaluate the effects of morbid obesity on gene and protein expression levels of RIP140 in visceral adipose tissue. 5 lean and 12 morbid obese subjects were used in the study.
This study observed a lower gene and protein expression levels of RIP140 in obese subjects. These results disprove our hypothesis, suggesting that an alternative source, such as GNC5, for the explanation of an impaired exercise-mediated increase in PGC1 alpha among obese individuals.